Today I'm going to talk about three key topics. I'm going to remind you what is an epithelial and talk about what is this. An apical and basal polarity. You remember apical will be here, the A, and basal will be in the bottom here. And if you see each one of these color structure, the ones that are in purple, the ones that are in blue, the ones that are in green, some of them are surrounding the cell, some of them work like a belt, other ones only in points. Those ones are proteins, so we're going to talk about that. Then I will tell you that those are called junctions, and we're going to talk about 4 cell junctions between cells. So I will show you this diagram multiple times. Today we're going to start talking about the first one, and I'm going to tell you the type of interaction that they have, the structure. So I will start zooming in and going from something very general to the different proteins that they have, and I will tell you about the functions. Okay, so let's start. And to start, I think this video summarizes all the class. And I also put it there in case that you want to study later.
Tissues are joined together by intercellular junctions that are located between the adjacent cells. Most types of epithelium and some types of muscular and nervous tissue contain intercellular junction. There are 5 types of intercellular junctions: tight junctions, adherens junctions, desmosomes, hemidesmosomes, gap junctions. The tight junction is an area where the plasma membranes of adjacent cells are fused together. These junctions protect underlying tissues from harmful substances by restricting the movement of substances, like hydrochloric acid, in the spaces between cells. Tight junctions are found in tissues lining the surface of organs and body cavities, such as the stomach, intestines, and urinary bladder. Adherens junctions are created when transmembrane proteins, anchored to a dense plaque of proteins inside the cell, connect the adjacent cell membranes together. Additionally, microfilaments extend from the plaque into the cytosol of each cell. The dense adhesion belts help resist separation of epithelial tissues. The desmosome is similar in structure to the adherens junction, except that intermediate filaments extend into the cytosol of each cell. These junctions help provide stability to tissues and are abundantly found in the epidermis of the skin and between the muscle cells of the heart. The hemidesmosome, or half-desmosome, provides strong attachments between cells and other extracellular materials, such as the basement membrane of epithelial tissues. Hemidesmosomes are most abundant in tissues that undergo constant frictional or abrasive forces. The gap junction is an open communication channel. Transmembrane proteins, called connexons, join together to create tiny tunnels that form gaps between the adjacent membranes. Gap junctions allow ions and other small molecules to pass freely from one cell to another. Gap junctions also allow electrical impulses to pass rapidly from one cell to another. Gap junctions are particularly abundant in some types of muscle tissue.
Okay, so I think that video, I put it there for you to study, summarize like an hour and a half of class, and it has information. So in case that you wanted to watch it later, you can watch it and it has all the information. So what I'm going to talk during the class is something like this. I'm going to start very general and showing you that we're going to talk about, here you have epithelial cells, and here these are the junctions that we have. I'm going to talk about four. These two that you can find here, this one are between cells, this one is the cells and the extracellular membrane. And those ones, we will talk about that if you're interested in that, in cancer biology, because they are very important for the migration to another tissue, yeah? So this is how they escape. So this focal adhesion and hemidesmosome are not going to, those one is not going to be in the test. If you want to know more about that, you can take cancer biology. So then I will start zooming in and telling you that this is not a circle or a square yet. We simplify that. It's a multi-complex of proteins. It's many proteins that work together to hold the cells together to perform different functions. So gap junctions are the ones that are found in the neurons, and as in the video they described, they are for cell communication, while tight junctions that we will talk in the next class help the cells to hold together very tightly. So they form a selective permeability barrier, okay? So this is how the class is going to go. And now I'm going to tell you remind you what we did in the beginning of the semester, that you were studying different type of epithelia. So you studied epithelial of the hands, and I didn't want to get into so much detail about all the names of the tissue. I just want to show that they were very different. Here they show that the epithelial can do a lot of things. So for example, they can form a tube, yeah, and later we will talk about at the end of the class, we will talk about how tubes are very important for us, yeah? So for example, the spinal cord comes from an epithelium. Most of your papers, we're talking about epithelial, and they talk about junctions, and they talk about the cytoskeleton. So this tissue is very important, and in this tissue, if you see, they are like bricks that have cement between the cells. Yeah, here they are all together forming contacts. And this is how multicellularity appear, and epithelial, they just form these complex structures. Okay, Jenna, super easy question that maybe you remember. What is the apical and basal polarity in an epithelial cell?
Is it A?
Yes. So apical. So when we talk about apical and basal polarity, here you can see two types of tissues. On top, you see an epithelium, and here you see a connective tissue. Yeah. So if you see, they look very different. While the epithelial cells are close to each other and they are in contact to each other, yeah. Here, the connective tissue is separated, and here what you find is extracellular membrane. Yeah. The SCM, yeah, the extracellular matrix, sorry, the extracellular matrix. And these are some fibers. So here you need to have all these junctions like that they have there. And here you can see, you have on the top the apical region, and here the lateral region, and here the basal region, okay? Below that, you have the basement membrane and the extracellular matrix. The difference between apical and basal is the look, you can differentiate. You can find in the apical a specific complex of proteins, and in the lateral, basolateral, you will find other complex of proteins. Okay, so remember that the epithelial, different from the connective tissue, is polarized. You have an apical region and a basal region. Apical here, basal here, and the connective tissue will be loosely organized. So you might remember the blood cells, yeah, that they are not they go individually. And if you're, for example, your bones, yeah, that you observe in some of the classes that I show you with the phase contrast microscopy, you saw them together. It was because the cellular matrix have a lot of calcium, but it was the cellular matrix was here and the cells were separated. Okay, so remember apical basal polarity, apical top, and at the bottom will be the basal. Maddie, what is a cell junction? Yeah, so perfect. It's a specialized structure that connects adjacent cells and facilitate the communication and adhesion. So here you can see different type of junctions. So the tight junction, as you watch in the video, they form like a belt. Yeah, they are surrounding the entire cell. So you see it in green, you see it in blue, something similar with the adherent junctions, while the gap junctions are like buttons, yeah, that they are located in a specific region and they have a thread. So these are localized, those ones are continuous. And the same thing happened with the desmosomes and with the hemidesmosome and the focal adhesions, okay? So you have two groups, some that are like a belt and other ones that are localized. And these adhesions, these junctions are very important to form these structures and allow the cells, the epithelial to create multicellularity and form all the organs. So yes, so definitely a junction is a specialized structure, multi-complex protein, yeah, machine that connects adjacent cells. Okay, so we finished the first part. Just it was to remind you, you already should know the difference between an epithelial and the connective tissue, and the presence of apical and basal polarity is something that hopefully you remember. And there are some structures that are called junctions, and we're going to talk about 4 junctions in this class, that they will be connected adjacent cells. Tight junctions, adherent junctions, gap junctions, and desmosomes. So Zia, there is not a perfect answer for this. I just want that you observe. Here you find the junctions. This is one cell. Here there is a membrane. Here there is another membrane, and this is the other cell. Yeah. What are the similarities between the 4 type of junctions? What do you think that they look similar in which sense?
All of them look similar?
What do you think what they have in common when you see them?
Not so sure.
Felicia, what do you think that they have in common when you see them?
Maybe the direction things travel.
So in all of them, you see that they start in the cytoplasm, they go to the membrane, and then they join in the extracellular space, okay? So all of them will be very different, look very different, but all of them will have 3 components. One that starts here close to the cytoskeleton, another one that goes into the membrane, and they actually fuse here, okay? So that will be a similarity, and I will be repeating it all the time. Except for the gap junctions, all of them will join with the cytoskeleton. And of course, the tight junctions will have here, you can see the components, and later we will study them. They have different type of proteins. The adherent junctions that are the ones that we will talk today, they will have these proteins that there will be questions, coherence, that need calcium to work. We have already talked about gap junctions before, and the desmosomes that we briefly will talk today. And this one will have very weird names that start with desmo something and desmoplakin and things like that you will see. Okay, so let's talk about these 4 things. these 4 junctions. Your book summarizes everything in a beautiful way. So for example, the adherent junctions and the desmosomes, the main function is the adhesion. And as I said, the adherent junctions will be like a belt, so we'll have a continuous zone, while the desmosomes will be only localized points. And here, so here you see the connection with the cytoskeleton, a linker protein in green, and here actually the core of the junction. Here the same thing, you will have the cytoskeleton, linker proteins, and the core. So here are this one are the adherent junctions, here are the desmosomes. The desmosomes will have intermediate filaments. The adherent junctions will have actin, okay? And if you see, other ones will also have intermediate filaments, and our will have actin, and we will talk about that later. Now, do you remember what is this plasmodesmata and where it's located? Somebody remember what is the plasmodesmata? Where it's located?
Is it like the gap junctions or like in plants?
Yes, exactly. So, I'm sorry if you're interested in agriculture, plant biology. Most of the courses of cell biology, developmental biology, all of them are oriented to animals because of the applications. So, generally in these introductory courses of cell biology and developmental biology, all of this is related to animals and not to plants. And these are one of the few slides that we have about this type of structure, for plants. So here you can see a vacuole. Here you see the nucleus. Guess that this is the nucleus, the cytoplasm, and here you can see the plasmodesmata. So they have these pores that allow to move some structures. This is not only happen in plants. In fungi, you can see one nucleus moving to a completely different part, and not on one but all of them, and it's just very weird. It's like when I think about this. So in your MCAT, are plants, animal, and fungi multicellular? Yes. Definitely. However, multicellularity is not as simple as we thought. Like, so imagine animals are like buildings that every apartment has a door, and you're not supposed to go to the apartment of somebody else. The multicellular in plants and fungi is like having a mall, being in a mall that everybody can go to one store and go to another one and live there, because you can have these spaces to move things. So all of them are multicellular, but the multicellular is different. And in the future, studying fungi is amazing to talk about genetics and cell biology of them. So again, plasmodesmata are found in plants that are intercellular pore that allow the membrane and cytoplasmatic continuity. And they are used for intercellular trafficking, communication, and signaling. So here you can see even in more detail what I told you multiple times. Cytoskeleton in all of them. You have linker or adapter proteins, and here actually the core protein that is like a zipper that allows them to get together. Some of them will have actin, some of them will have desmosomes. And this is how these proteins hold together the two adjacent cells. And today we will talk about these ones that are the adherent junctions. And here, this is just an introduction about adherent junctions.
The human body is made up of trillions of cells that form tissues and organs like the heart and liver. If our cells weren't able to stick together, our organs wouldn't be able to do their jobs properly. Two scientists named Takeichi Masatoshi and Rolf Kemmler discovered that cells stick together by producing a protein called cadherin. There are many different cadherins, and they each help cells that belong to the same type of organ stick together. Cadherins make connections with the rigid inner part of the cell, called the cytoskeleton, to help stabilize their contacts. This is actually mediated by another family of proteins called catenins. Some forms of cancer are harder to treat if cadherin isn't working properly, since the cancer cells can lose their stickiness and escape their tissue of origin. Continuing to study how cadherin works and how it's involved in cancer could help us develop treatment strategies to get people better, faster.
Okay, there were more complicated videos. But I found this one, I like this one because it was very simple and silly. And now we're getting to something more, a little bit more complicated. So they were talking about coherence. So let me see if this works. So E comes from epithelium. Yeah. Yeah. So here you have the E coherent, that is the epithelium coherent. And here you have in the outside of the cell, in the extracellular space, you have this protein that goes from here to the transmembrane domain and go to the cytoplasmic domain and connect to all these three proteins that are called catenin, okay? And then they connect to actin. And we will talk about how they fuse and all the functions in a second. And we were there in the back. Could you help me? What is the primary role of the coherence in cellular biology?
Like the C.
Mediating calcium-dependent cell adhesion. Yes, yes. So I don't know if in the video they talk about that. I think in the next video, this is very important. The coherent to work. If we remove the calcium, they will not attach to each other. So perfect. So coherence, they interact in different ways. So for example, you can have homophilic or heterophilic interactions. So here you can have the two blue proteins that they could be the same and they will be homophilic, and you can also have heterophilics when the proteins are different. And even in a single cell, you can have the both types. So when you have these adhesion molecules, you have some that are homophilic, that they have identical receptors, another one that they have that are heterophilic, that they have different receptors. And yes, so they are mediating calcium-dependent adhesion, cell adhesion. Okay, so here you can see another diagram, and you know that when we talk about adherent junctions, we have actin always there. Yeah. So here you will have these brown three proteins that are called catenins, and here you will have these CAMs that are cell adhesion molecules, and you have two main that are coherents and neptins, okay? So this one is one of the simplest one. And remember, this, you will find this term, CAM, that is cell adhesion molecules. And these are two examples. I haven't told you anything about this protein that is called adherin. Those ones, so, oh, sorry. Again, same idea. You have the core here. Then you will have linker proteins that in this case are the coherent, the catenins, and you have 3. And here you will have the cytoskeleton that in this case is actin. And in all the junctions, except for the gap junction, you will see core, linker, and cytoskeleton. I'm just going to tell you about this, but if you go and study this in more detail, you will see that there are additional proteins, and every time they find more proteins. So for example, this adherin helped to stabilize these cell adhesion molecules. Okay, and this one you need to remember for the test, and actually for one of the questions, the function of these linker or adapter proteins, that they fuse the core to the actin. Okay, so if we're going to start talking about, based on your textbook, there are 2 types of adhesive junctions, the adherent junctions and the desmosomes. And the adherent junctions are intercellular attachments that join the coherents to the cytoskeleton. And here you can see again the same things that I have told you. Okay, Angus. So what is the primary function of the alpha-catenin?
B.
Be linking coherence to the cytoskeleton. I think you're right. So, perfect. So there is a lot of catenin, beta-catenin, alpha-catenin, P120 catenin, and there are more proteins, but yeah, here you find the actin in red attaching to the alpha-catenin. Okay, so if you want to read the textbook, they will tell you more information. Here you can see the picture of a desmosome. This is the electron microscopy picture, how they get the colors. This is just with a software that allows you to see, to make it easier to see how a desmosome will look like. This is a diagram that you will find, again, the same structure that I have been telling you. There is a core here that are the connecting filaments that have weird names that we will talk in a second. There is a plaque here, a cytoplasmatic plaque, that it will link these core proteins to the cytoskeleton. In this case, the cytoskeleton will be intermediate filaments. And if you remember the video at the beginning, these are localized, they are not forming a belt, and they are in location like your heart, that they form very strong connections. Okay, core, linker, and cytoskeleton. Again, your textbook is great, and they show you a lot of information in case that you want to study. For example, in the desmosome, the associated structures are called tonofilaments, that are just a type of filaments that have keratin. Do you remember? If you remember the disease that unfortunately, that mutation that we watched last time, that people develop blisters, a lot of blisters, is because they have a mutation in the keratin. So the desmosomes are not working properly because there's a mutation here in these structures. Okay, so I just told you about the importance of the epithelium, and the epithelium, as they are together, they have junctions, and there are 4 types of junctions, and all of them have the same structure. And I start talking a little bit about adhesive junctions. And now I'm going to show you that although we focus on coherent, that as Derek said, they have calcium or Page. You two say the same thing? Yeah. So they have calcium, and this is very important. And I told you they have a transmembrane domain, an extracellular domain, and a cytoplasmatic domain. Here you can see the desmosomes that I didn't get into detail, but it's the same idea. Transmembrane domain, extracellular domain, and cytoplasmatic domain. And here you have other type of adhesion molecules. All of them have a small variation in the number of segments, in the way that they connect to other cells, and they have multiple functions that we will talk later in the following class. Okay? I think Asher, could you help me with this one? What are desmosomes?
B.
Yeah, so pretty easy. Super strong cellular structures. And we saw that they have ton of filaments that are specific type of intermediate filaments. Here in your textbook, they summarize all the things that I have said, and you can study it from there. I just put it there in case that you want to see it. And they have more structures. Okay, so we finished with the epithelia. I'm sure that you know about this. Now you know that we're going to talk about 4 types of junctions, and that they have a core section, they have a linker proteins, and they have a cytoskeletal connection. But all of them, this one have different proteins. And now we're going to start with the adherent junction. And here you already know all this information that I have been telling you, that they mediate, they are coherent-mediated junctions, that they interact with actin, that they are located especially in the epithelial, and they rely on coherence mainly. Okay, so they are located here. So here you see the tight junctions. This is the apical region, and in the subapical region, you can find the adherent junctions forming, again, this belt of adhesion. Here they will tell you a little bit more about the details of coherence.
After its translation from mRNA, the classic cadherin protein is transported to the cell membrane. The cadherin establishes itself as a transmembrane protein with distinct cytoplasmic and extracellular domains. As seen in this conceptual model, the extracellular segment, which is responsible for the cadherin's ability to link adjacent cells presenting cadherins of compatible type, includes 5 characteristic domains. These cadherin domains interact with domains on neighboring cells in a calcium-dependent manner. In the presence of calcium ions, individual cadherins pair up to form dimers on one side of the intercellular junction. These cis dimers then bind with other dimers on the opposite or trans side. When calcium is removed from the environment, the cadherin molecules fail to bind, and cell-to-cell adhesion is disrupted. The cytoplasmic tail remains within the cell and forms a complex with a trio of catenin molecules, P120, alpha, and beta-catenin. This molecular complex links the transmembrane cadherin protein to the actin cytoskeleton. The cadherin-catenin complex is also important in cell-to-cell adhesion, as cells lacking catenins or expressing cadherins without catenin-binding domains fail to adhere as normal.
Okay, so this summarize a lot of questions. Duncan, here they are talking about how many extracellular coherent repeats are typically found in the classical coherence. You might, oh, you have nice mustache. I just realized. You might not remember this question, but later you will remember for the test and the exam. And I think I pointed the answer unconsciously. So how many they have?
I think it said 5.
Okay, and I am not good at poker. I will be the worst poker player. So yes. So here you can see 1, 2, 3, 4, 5. Yeah, and those are the extracellular domain. Again, sorry to repeat these things, but I think this is something that I want you to remember. There is a transmembrane domain and there is a cytoplasmatic domain. Okay, so this is how normally the coherence, the classical coherence, the structure of the classical coherence. And again, I have been telling you all the time that this is like Lego, that you have some units and you change a little bit the components and you have a new protein, yeah, or you have something new. You have a lot of diversity of coherence that it could come or from the extracellular domain or from the transmembrane domain or from here from the extracellular, transmembrane, or cytoplasmatic domain. And there is just so many of those, okay? So here, what we're talking with Duncan is that there are extracellular repeats that they can vary from protein to protein, as you saw there. So you can have, in the following slide, I will show you that they can be more domains, and there is a lot of variation on the repeats, or you can also have a mutation with a different one. Yeah, so you have different colors in those cases. So here is the transmembrane, and here is you have a lot of variation in the cytosolic ends. Okay, so I think, Duncan, hopefully I don't do the same thing in the final exams and do that, but good. So here is what I was telling you. There is the coherent structure, and if you see this is the classical that we were talking and that we watched in the video, but if you see there is a lot of them, yeah? And again, this is like a Lego game that you can add more components, so you can have small variations and create different things. Here, this flamingo, if you see, is a little bit weird because the transmembrane domain goes over multiple times in the membrane. Okay, and here you can have all these different, the legend of that. I'm not going to ask you in the test. I might ask you about this one, yeah, but I'm not going to ask you how many they have in the protocateran, the fat. Yeah, I'm not going to ask you this structure. I just want that you know that they exist and how they vary. So all coherents possess calcium binding extracellular repeats that they vary in numbers. The non-classical coherents, they can have additional extracellular motif, like here the EGF motif, or in flamingo that you see here. And in flamingo, as I said to you, they have this very weird transmembrane domain that go multiple times over here, over the membrane. This is something that you also watch in the video about this connection. So between the two cells. So you have two types of connections, but remember the E coherent, that is the epithelial coherent. So this E, it will be super easy. You remember that? And scientists, at least in this case, they didn't try to make life easier. So if you see an N coherent, it will be neural coherent. If you see a P coherent, it will be placental coherent. Yeah, so that makes a little bit easier your life. Yeah, and here, so as you're watching the video, you can see that this is like a zipper, of your sweater. Yeah, that they are complementary, and they can be connections that are in cis in the same chain, or they could be connections trans, or they could be both. Okay, and you watch already that in the video, and this is what I was telling you about the zipper, that they interact like in a zipper mechanism. Okay, so so far, just to summarize everything that we have talked, I have told you in the beginning that there is an intracellular domain, a transmembrane domain, and extracellular domain. And here you will have a lot of proteins. Here they will be catenins. Here in the when we study the E coherent, that is the epithelial coherent, you can have a cytoplasmatic, a transmembrane domain, and here all the repeats, and they need calcium. Same thing, we will talk something similar. We will see when we study other the tight junctions and the desmosomes. Now I'm just going to go very quick. So the beta-catenin, that is that one. It will bind to the tail of the coherent, so you can see the cytoplasmatic tail, and that one will bind to the alpha-catenin. Why we care about that? Because this is very important for cancer formation. Yeah. So why we're going to disassemble the junctions? When do you think that it's important to disassemble a junction in a tissue? That you do right now, your junctions are disassembling, and the other ones are forming for a process that is very important. That is called apoptosis. Every time that a cell dies, they have to disassemble it and to go inside and die. Yeah. The problem is when they don't die in the right way, yeah, they can go and form cancer. So this is why in the video and at the end of the class I will talk about that. So you have this P120, and you can study this for the test that they attach to the cytoplasmatic tail. So here is the one that is, I try to color code it for you. You think that they attach to the tail of the coherent, and it helps to regulate the coherent, to stabilize it, to regulate it. And I need to talk about rho in the following class, in the following classes. Okay, so I told you the structure of the coherence. I told you all the components that they have repeats. That now I'm going to talk about some of the functions. Why coherence are important? So here we have a diagram of a bacteria that is in green, and here you can see all the different complex adhesion molecules that are there. And of course, bacteria are great at using all the things of the cell to try to get inside of the cell. So there are many people that study how bacteria are able to use this mechanism to get inside of the cell. So they have complementarity here to those molecules, and they end up getting inside. Yeah, so many pathogens use this adhesion system to get inside. And there is a lot of work in that. Another important is that for tissue specificity. So if you want to take developmental biology, we talk a lot about this. Here you start, and I think Angus have these sections. Some of them belong to Angus' paper that he's going to have in his poster. So here you can see an antibody staining, yes, expressing P coherent, and in green the one that expresses the E coherent. So they have different stickiness. So later during development, they will separate. They will self-organize. They will not be attached to each other in the correct way. Yeah, and of course the E coherents are the epithelial coherents, and these P coherents are the placental coherent. Okay. So here, this is another very important diagram, something that here I'm just going to talk 5 minutes, and maybe in anatomy and physiology also there's like 2 pages of this. In developmental biology we'll talk about for 2 weeks about this. So in epithelia you have the expression of a different coherent, and now you see that the entire tissue went inside. Okay, so you have one type of coherent, you have expression of a different coherent in the nucleus, and now it disassembled why we went into so much detail talking about this? What is the implication of this? The application that this process of forming a tube have in human biology. Piece of a tissue that change and then is able to migrate. Yeah, so this is important for cancer biology. This is exactly what happened during cancer, that you disassemble the adhesion molecules and they start traveling into a different way. Yeah, so this is also important in developmental biology because this is how you form the neural tubes. This is important for evolution because frogs, chickens, mammals, they go and do the same thing, but the spinal cord is all forming a different way in each organism. Okay, so remember, the amount and the type of coherent is very important. Switching a different type will produce a different tissue. Here in this case is the neurulation, that is the form of the neural tube. And here, pretty easy, you go from an E coherent, epithelial coherent, and you change to N coherent, that is the neural coherent, and you can form the neural tube. This also processes the sorting, that is also very important. So if you press different coherent, they self-organize and they form different type of tissues. Okay, and this is a mutation, this is an embryo, and this is a mutation, that lack of coherence, so the cells are not able to attach properly. Embryos that lack of coherence, they died very early in development. This is very difficult to study coherence later because it makes a huge mess in the cell. Okay. So this is going to be in the test. EMT, yeah, that is epithelial mesenchymal transition. So the cell that you saw going inside, yeah, the neural tube, the neurulation, yeah, it's a process called epithelial mesenchymal transition. So I start the class telling you, do you see the epithelial is very different from the mesenchymal tissue, from the connective tissue. Yeah, there is a way that you change the genes, and it's very common that you can transfer, you can go in that way, the normal condition. However, cancer can use the same thing to expressing the wrong genes to turn into to migrate.
This narrated animation shows how cadherin proteins on the surface of one cell interact with cadherins on another cell. It also shows how different cadherins expressed on different tissues help to segregate tissues.
Oh, sorry, this is aesthetically.
Here, we start out with an epithelial cell with its villi extensions. The cell then acquires integrins, which help it attach to the basal lamina. Next, the cell develops its adherent junctions, which helps epithelial cells attach to one another. Following this, the cell develops its actin, and then its E-cadherins. The basal lamina appears, and then its apicopolarity complexes. Our epithelial cell is now complete and joined by two more epithelial cells to begin the epithelial mesenchymal transition. In step 1, normal epithelial cells are attached to one another through adherent junctions containing cadherin, catenins, and actin rings. They are attached to the basal lamina through integrins. In step 2, paracrine factors can repress the expression of genes that encode these cellular components, causing the cell to lose polarity, lose attachment to the basal lamina, and lose cohesion with other epithelial cells. In steps 3 and 4, cytoskeletal remodeling occurs, as well as the secretion of proteases that degrade the basal lamina and extracellular matrix molecules, enabling the migration of the newly formed mesenchymal cell. And finally, in step 5, the mesenchymal cell gains the ability to migrate and differentiate into other cell types. This gives rise to the formation of defined structures, such as the neural crest. After undergoing EMT, Here, we start out with an epithelial cell with this villi.
Okay, so this is a normal process that is that during cancer you can activate those genes that happen normally during development. This is how we all have our spinal cord when we in early development. Same thing happened here. You can take the same genes and mutate them and activate them to start to turn in to start growing and then to produce this epithelial mesenchymal transition that will, and so here you see AMT, and that later it can end up leading into cancer. So something that it looks that is not very important, like the coherent, like the seeping of these structures, have been extensively studied because they are very important to understand how to assemble and disassemble a tissue. And we know that in normal conditions, this is very common. Yeah, I told you, while we are sitting here in this very hot room, our cells are dying and are proliferating, and they are disassembling the coherent. When we were babies, we needed that some epithelial tissue turn into mesenchymal tissue to form other structures. What cancer is doing is not something new. It's just reusing the same genes to do something different. Then, if you remember the previous class that we were talking about how the crawling cells are taking place. So you have actin that polymerize and depolymerize. So they just have mutations in actin, and in that way, this is why cancer doesn't happen in 2 days. Yeah, it will happen in 10, 20 years. If you take cancer biology, you will see that studies have showed that after 20 years of smoking, you see that the common pattern is that people start developing cancer. Why? Because you need to go from all this type of mutation to take place to later be able to invade other type of tissue. What is MET? MET. So we have EMT, is epithelial mesenchymal transition. MET is mesenchymal epithelial transition. So the cancer, what they do is now go and invade another tissue, and they turn mesenchymal tissue and they turn into an epithelial, and this is how the invasion takes place. So this could be in the test. What does the mesenchymal the EMT stand for? So it's epithelial mesenchymal transition. Okay.